Welcome back, everyone, to Joyful Oral Path. Today, we begin our embryology series, which is essential for the understanding how the head, face, and oral tissues develop. A solid foundation in early human development allows us to appreciate the origins of dental as well as craniofacial structures, and also helps us understand why certain congenital anomalies occur. So, in this session, we'll walk through the basic events that prepare the embryo for forming the major tissues of the oral cavity. In humans, almost every somatic cell in the body contains 46 chromosomes. This is the diploid number. These chromosomes exist in pairs, 22 pairs of autosomes and 1 pair of sex chromosome. But when it comes to reproduction, the story changes. Germ cells, the sperm and the ovum, collectively known as gametes, must contain only 23 chromosomes each. This reduction is crucial because during fertilization, these two haploid cells combine to restore the normal diploid number of 46 in the newly formed zygote. The process that reduces chromosome members in gametes is known as meiosis. And while meiosis normally ensures correct chromosome distribution, errors can occur. When chromosomes fail to separate properly, the resulting gametes may carry too many or too few chromosomes. If such a gamete participates in fertilization, the zygote can end up with abnormal numbers, leading to conditions like trisomy or monosomy. A common example is trisomy 21 or the Down syndrome, which can present with facial differences, delayed dental eruption, and oral structural variations. Not all congenital conditions arise from chromosome abnormalities. Many result from the single gene defects. About 10% of human malformations fall into this category. In autosomal dominant disorders, only one altered gene from either parent is enough to manifest the condition. Examples include dentinogenesis imperfecta and osteogenesis imperfecta. On the other hand, autosomal recessive conditions such as certain types of microcephaly or cystic fibrosis requires both parents to contribute the defective gene. Understanding these patterns help us connect genetics to dental tissues and craniofacial development. Prenatal development is divided into 3 major phases. The first phase, lasting about 4 weeks, focuses mainly on rapid cell proliferation and migration. The second phase, which is week 4 to week 8, is the critical period of morphogenesis, when all the major organs and structures begin to take the shape. This is also the period when most of the congenital defects originate, since the developing embryo is extremely sensitive to disruption. From the end of the week 8 until birth, we enter the fetal stage, where growth and maturation becomes the primary processes. A key principle in development is pattern formation. How cells decide what to become and where to go. This is guided by 3 interconnected processes: induction, competence, and differentiation. Induction refers to the signals or cues that tell a group of cells to change their fate. Competence means those cells are able to receive and respond to those signals. And differentiation is the final outcome, where cells acquire specialized functions. Homeobox genes and growth factors play central roles in regulating these steps, including those involved in the tooth formation. Following fertilization, the zygote immediately begins rapid mitotic divisions. These early divisions do not increase the overall size of the embryo but instead create many smaller cells. By the third day, this compact ball of cells is called the morula. As fluid begins to enter the cell mass, a cavity is developed, transforming the morula into a blastocyst. This blastocyst stage is crucial because the embryo now has distinct cell populations that will carry out different developmental roles. Within the blastocyst, two important cell types emerge. The trophoblast forms the outer layer and plays a major role in implantation and placental formation. Inside the cavity lies the inner cell mass or the embryoblast, which will ultimately give rise to the entire embryo. This separation of cell lines marks the beginning of true embryonic organization and sets the stage for forming the germ layers. By around the day 8, the embryoblast differentiates into two layers, forming what is called the bilaminar germ disc. The upper layer, which is the epiblast, consists of columnar cells that line the developing amniotic cavity, while the lower layer, which is also known as the hypoblast, is composed of cuboidal cells that help form the yolk sac. During this period, the embryo also establishes its head to tail orientation. A thickened region known as the procaudal plate marks the future head end and plays a role in early body axis formation. During the third week of development, the embryo undergoes a major transformation called as gastrulation. This is when the bilaminar disc becomes trilaminar, containing three primary germ layers. It starts with the appearance of the primitive streak on the epiblast surface. Epiblast cells migrate inward through this streak, pushing aside the hypoblast to form the definitive endoderm. Additional migrating cells spread between the layers to form mesoderm, and the remaining epiblast becomes the ectoderm. So these three layers will generate all the tissues of the future body. At the head end of the primitive streak lies a small depression called the primitive node. Cells that pass through this node migrate forward to form the notochordal process. This structure then develops into a notochord, a stiffened midline rod that serves as the primary axial support for the early embryo. More importantly, the notochord sends signals that initiate the formation of the nervous system and guide early patterning of the head and spine. As more cells migrate through the primitive streak, they form two major new layers. Those that displace the hypoblast becomes the true embryonic endoderm, which will later give rise to the lining of the digestive and respiratory system. The cells that remain in between form the intraembryonic mesoderm, which gives rise to muscle, bone, cartilage, connective tissue, and blood vessels. The original epiblast cells that stay on top becomes the ectoderm, later forming skin, the nervous system, and the enamel of the teeth. To summarize, early embryonic development begins with rapid divisions of the zygote that form the morula and later the blastocyst. The inner cell mass organizes into bilaminar disc, establishing early body orientation. Gastrulation transforms this disc into a trilaminar structure containing the ectoderm, mesoderm, and endoderm, which are the three fundamental layers from which all the tissues, including those of head and oral cavity, arises. These events set the foundation for the craniofacial and dental development we will explore in the upcoming videos. Thank you for joining me on this journey through the early embryonic development. If you enjoyed this video and want to continue learning about craniofacial and dental histology, don't forget to subscribe to Joyful Oral Path. Our next episode will dive deeper into the next stages of embryonic development and how they lead to the formation of the head and oral structures.